My name is Valentina, and I'm one of the co-founders of Pytri AI. I wanted to start by saying a massive thank you to each and every one of you for being here today and taking the time to join us. It's truly amazing to see some familiar faces as well as some new ones. Our common passion for innovation is what brought us here today, bringing together a stellar lineup of speakers from academia, industry, and our own network of collaborators. We wanted to create a space where ideas could collide, where expertise could be shared, and where we could all explore on the incredible potential of lab automation and biotechnology. So today I wanted to introduce you two other integral co-founders of Pytri, my sister Michelle and our CEO Ali. So before we're going to dive into the heart of today's event, I just wanted to take a short moment to reflect on a journey that's been nothing short of extraordinary for us. So recently, we actually celebrated Pytri's 1-year anniversary, which was a moment filled with joy, excitement, and a lot of pride for us. And this really brought us back to reflect on how everything got started for us, which was actually the molecular biology lab as every student doing research in university, where we bonded over having found some tasks pretty inefficient, specifically bacteria cell counting. And I'll let Ali dive a little bit deeper into how we got here today.
So these were the days before ChatGPT took over our lives, before we started automating everything with ChatGPT, other large language models. And so this was back when we were still coding solutions like ImageJ, which was our competitor at the time. And so we had to go there and try and figure out how do we annotate biological entities, how do we determine what a colony looks like. And so we were looking at these colonies and trying to delineate them using color, using lots of techniques that worked great in our lab. But once we took that out of our lab and put that in another lab, it wasn't able to recognize colonies anymore because lighting changed and all of that. And then, we joined District 3, which is where we enter the competitions as PhD students. We weren't PhD students at the time. We entered in that competition and we were able to build an AI solution that was able to detect colonies.
District 3 also gave us a very supportive ecosystem which helped us expand our business model, expand our network. It really helped us understand who we are better as researchers, as people, and as business owners now. So that was a very important moment for us, definitely.
And so a lot of the aspects that we focus on when we're looking at biological data in terms of large language models is what do we do with the data that we get? How do we analyze it? How do we interpret it? So think drug discovery. That's what we're focusing on, and discovering novel drugs using AlphaFold, using all these techniques in terms of analyzing the data that we're getting. But there's a huge bottleneck, and that bottleneck is actually obtaining that data. So it's really difficult to obtain that data in terms of labs are producing enormous quantities of data that sometimes sit on drives and are just really inaccessible, and there's a big throughput issue. So our approach and what we were looking into was trying to automate that sense of, how do you automate the image taking and then the analysis of those images before we get that raw biological data that language models are now interpreting and creating models of how things work in terms of AlphaFold, etc. And so that's a large part of automation.
Yeah, and another important thing we had to take into account at that same time we were developing Pytri. Me and Valentina had been working in a COVID lab at the time, and we all know the pandemic took us kind of by surprise. And we were working in a RNA extraction lab and qPCR, and a problem that a lot of smaller labs had was scaling to be able to mass produce these tests. And we saw with our own eyes and hands how in the beginning we would extract RNA and it would take hours, 5-8 hours, and it would take so long to do these tests. And especially as a public health matter, it was really important to get those results out really, really fast.
And the pressure was high.
Exactly. And then we ended up acquiring different machinery that was able to extract the RNA faster, do the PCR faster, not have that many people in the lab at the same time, because that also was a big problem, having a lot of people in close proximity to each other. And then we ended up doing maybe 1,000 tests a day with just 2-3 people in the lab.
Compared to 100 before.
Exactly. So that changed a lot for us in that space. And that's what mission we were trying to bring at Pytri as well. Small scale labs actually benefit from this a lot, to be able to just modify based on their needs at the moment.
Yeah. So that is a large issue in terms of small labs not having the budget to buy a $20,000 colony picking machine or a Petri dish analysis tool. There's a lot of automated tools out there, but are prohibitively expensive for smaller labs. So what we do is we rely on student interns or student volunteers that do these menial tasks
and
ourselves, which we all did. But with AI, with large language models, all of that is changing in terms of analyzing this data. So an aspect that you can think of is, okay, why don't I ask ChatGPT and I take my phone out and take a picture of a Petri dish and ask it to identify bacteria, which a lot of labs have tried doing, but they face struggles in terms of ChatGPT is trained on the entirety of the internet. It being able to precisely identify and count objects on an image is an incredibly difficult task that doesn't work with generalized AI models, maybe one day, probably one day. But for now, you need specialized AI models that are trained on these biological data sets that are usually locked in Google Drives of lab researchers. And so it's difficult to get that out and then build this into an AI model and then actually release that to the public. So that's the bottleneck that small labs are facing in terms of they don't have the time, energy to build these solutions.
Which is where we come into play, and this is something that we thought of, is essentially providing custom solutions to small labs, big labs, to essentially anyone that has a database that needs it to be counted accurately and efficiently. We are able to essentially just target our model to what their specific needs are. And another big addition to our platform that was recently released was the ability to return comprehensive reports, which is something that can save so much time in terms of just returning the data and having it in an organized and very clear way. So these are just a few examples that we've thought of in terms of automation and how our solution is automating lab work these days. But we're gonna hear a lot more on that soon. So yeah.
Because the big trend, let's say, of nowadays is automating every aspect of our job. So that doesn't only include lab automation, but also the tools that we use to communicate ourselves. Microsoft is huge on automating things with Copilot, automating your emails, building on all the data you've been accumulating. It learns from all the emails that you've ever written in terms of generating and helping you out. And so it is a big aspect that's taking part of all of our lives. The big issue with that is also data privacy and data security. So your lab information, all of that, are you keeping it within Canada? That's a big issue when you go and talk to hospitals. Your data has to remain on servers that are located sometimes in the same province as you. And so is that data staying there? Is it going out? If you're using Microsoft Copilot or using ChatGPT, that information is being sent to servers in the US. Does that align with your company's policy? That's another big issue that companies are facing nowadays, as they do seek to automate everything while also skirting around lawsuits. And that's right before we automate the legal system with AI lawyers. We're not there yet. We're very soon. There are AI lawsuits that are being filed as we speak, and so that's a little rabbit hole that we can delve into another day. And so
I think it's a good example of basically just showing how you can automate your everyday life, but also to still kind of have a clear head around it and still guide yourself and to be the one that's in charge of it. But we're not going to go into the ethics of AI, that's not today's purpose. It's just a fun little fact to think about as we're here.
And I guess it's time to meet our speakers for today's event. I would like to invite John Drucker from BME Montreal to tell us a little bit more about the productivity being the new trend in the industry.
Thank you. everyone for presenting me. Thank you for having me, Ali. It's great to see you. My name is John Drucker. I work at PME Montreal in West Island, but I've worked with other ones. And for those of you who don't know what we do, we are an arm of the City of Montreal that helps with funding for small startups in different areas, and we have a focus on innovation, obviously. And yesterday, and the day before, I was at the World Summit on AI, and so much was talked about that touches what we're talking about today, which is huge troves of data that are going to be unlocked, and they need to be examined to be able to basically give the innovations and give the forward progress that we need for society. And when I met Ali, it's almost a year ago, I guess. Yeah. He explained to me what the issue was with colony counting, and I didn't know, but this issue of having to do things manually, to me, I thought, wow, it's 2024, isn't AI taking care of this? And Ali explained to me, he said, no, we still have people doing it manually. And it was a shock to me because I figured this is the type of thing that's ripe for this, and there's so much data. And then, as I started to understand the problem more through Ali, thank you again, and the team, I started to see where this is all coming together in terms of productivity, in terms of giving us solutions to issues. So drug discovery, looking at how things are working, and then taking that and putting it into a working process. And if you have been following the news the last little while, the government of Canada and Quebec has been talking about the lack of productivity we have in Canada. It's an issue. Quebec, among the major provinces compared to Ontario and compared to British Columbia, is even further behind. And in the position paper that was issued 2 days ago, they spoke about a 12-step plan to try and advance practical applications of AI and research to increase productivity in all aspects of our lives, science being one of them. And it is acutely aware to so many that we have this treasure trove of data that we know we can now start to do things with, but how do we do it effectively and efficiently? And that's why we talk about productivity. And we're not talking about how many widgets can I make, but what can I extract from the data efficiently using what tools are available to me, AI, other things as well, high-powered computing, and try and get the message home to people that you have a brain and we should be letting the AI do the heavy lifting. Yes, there's work to do, there's prep to do, there's getting the data out, and we're at that beginning stage. But once we start to figure this out, and we start to get tools like Pytri, like other ones as well, all of a sudden, we can look at things differently. And we can actually become more productive and use our brains on the creative aspects of our brains. And this is where people say, I have to spend so much time filling out paperwork or doing these kind of things. I hate it, I do it too. But it's now being able to say, stop, and now I have information in front of me with some knowledge behind it, with some data behind it to make better decisions much more quickly. And it's that time to market, it's that time to a result, it's that time to pushing forward research is what not just Montreal cares about, not just Quebec, Canada. This is a general concern across the globe of how do we get things to market more quickly. And productivity is at the bottom line. And it's doing it well, doing it quickly, but being able to repeat a process and separating out the automation side from using your brain side. What can I do? And Ali, you're a great example of this, and so is your team, of how do I approach this problem differently? And for me, it was kind of like, isn't computer vision something that we could be doing with AI? Yes, we can. And for so many different areas. There are other aspects of AI, and to be honest, I don't think ChatGPT is where it's at. And my opinion after going to the World Summit yesterday was, we're going to be needing smaller language models, more defined ones, so that we can, instead of sifting through all the stuff, getting to the point of where we need to get to quickly. So I think you guys are on the right track. It's nice to see. So when we talk about productivity, it's not some nebulous thing that economists only deal with. It's what we all deal with when we're stuck with, oh, I've got to draft this manually, I've got to do this manually, and you could be doing something more value-added. And this is where I think Pytri has a great example for all of us in the island of Montreal, as well as beyond this province. These are the types of innovations that we want to push. These are the types of things that actually make a difference. It's not another music generator, because there was one of those there. If we could do artificial AI music, I don't care. What I care about, or what we care about, is seeing what are the things that impact us day to day, and being able to push through these innovations that can get to market and have value for small, medium, large-sized companies who haven't even thought about this, or if they have, they get lost in the weeds. Pytri comes to market with something that is concrete, useful, and ready to go to market. That's what's so important. That's what makes you guys different, is that you've got something that people can use now. So you should be proud, and congratulations.
Thank you.
So thank you for having me. If you guys have any other questions, please let me know.
Thank you so much, John, to this insightful presentation. Now, if we can continue with Donald McLaren's presentation.
Hi, my name is Donald McLaren. Thank you, Ollie and the Pytri team, for the invitation to speak with everyone today about AI and neuroimaging. Behind me, you'll see two examples of MRI scans from my daughter. The following are my own views and opinions. It is indeed an exciting time in the field of AI and image processing. In the next few minutes, I'll discuss brain segmentation as well as axon and myelin segmentation in preclinical animals. AI is in fact not new to neuroimaging. It's been around for over 20 years. Artificial neural networks were first used in the late 90s to segment MRI images of the human brain. Using MRI, we collect data from voxels or volume elements. These are just like a pixel, but in 3D. The current acquisitions that we have for MRI collect over 10 million voxels every time we scan the brain. And each of these voxels has a different intensity telling us about the properties of that voxel on that specific machine. Each machine gives us different absolute values, and as well as differences in the signal magnitude differences between tissues. You can think of this as two Gaussian distributions, where on one machine they might be this far apart between gray and white matter, and another machine they might be spaced further out. There are other complexities too, such as non-uniform intensities and individual differences in brain anatomy that make segmentation more complicated. In the late 90s, researchers from the Montreal Neurologic Institute at McGill, down the street from here, developed a multi-layer artificial neural network to classify the brain into gray matter, white matter, cerebral spinal fluid, or background. To do this, they included three spatial parameters, which are maps of the probability that a voxel would belong to a given tissue class. The three maps included gray matter, white matter, and cerebral spinal fluid. And also, the other input was one or more types of MRI sequences that have already undergone that intensity non-uniformity correction that I mentioned before. The result of this was an automated, robust, and accurate segmentation pipeline, which had several advantages. First, elimination of the time-consuming step of manual segmentation. Two, the elimination of the bias from manual steps and manual intervention in segmentation. And three, it increased the number of data sets that could be included in an analysis. It was advances like this that have enabled quantitative data analysis in clinical trials and population-based studies like the UK Biobank, which now contains over 60,000 MRI scans from people in the UK. Today, improvements continue to be made in the brain segmentation, and the field is always adding new features, such as super resolution, where you can take a low-resolution image and change it to a much higher resolution to find finer details of that image, all using deep learning approaches. Now I'm going to turn and talk about what we can do preclinically and going deeper into the brain. I want everyone to raise your right hand. While this is a simple action, many parts of your brain were involved, and each of these parts of the brain, as well as the muscles in your hand, are connected by cylindrical components of neurons called axons. These are only a few microns wide. Typically, each axon is going to be wrapped in a myelin sheet in a spiral pattern. This sheet helps protect the axons as well as speeds the nerve transmissions along the axons. In neurodegenerative conditions where the myelin is lost, such as multiple sclerosis, the nerve transmission can get slowed or even disrupted, which ultimately leads to the clinical symptoms that we've seen in that disease. In research, we can induce demyelination, creating white matter lesions just like we see in MS. And then we can assess the ability of drugs to induce or improve the remyelination process. Recently, Convelo Therapeutics has developed a high-throughput, unbiased, AI-driven segmentation tool to identify myelinated and unmyelinated axons in these lesions. They are then able to calculate the myelin thickness as well as the G ratio, which is a measure that reflects axonal function and integrity. Their unbiased approach has allowed them to investigate 1,000 times more axons than legacy approaches, and they've been able to evaluate the effect of their drugs on remyelination. There are many other examples in imaging where AI has reduced manual effort, such as breast cancer screening and detecting abnormal EEG segments for an expert human review. rather than asking a physician to review the whole continuous 24-hour EEG recording. In summary, AI in imaging reduces possible bias, maintains or improves accuracy, and increases images throughput. It is important to remember that AI algorithms are developed with a specific context of use, and those algorithms should only be used within that context and not for expanded use cases. With more data, algorithms can be refined to improve their precision and accuracy, moving us toward personalized medicine. Thank you very much.
We're going to continue with Benjamin Scott, presenting from the Global Institute of Food Security about engineering biology for food security, the role of automated labs.
So I'm going to talk a little bit about my perspective on lab automation in the field of engineering biology. I think I'm going to end up echoing quite a bit of what John mentioned in broad terms, but I wanted to give some specific examples about how we're using automation in the lab now, and I think the potential for AI in biology. So what is engineering biology? I think the simplest definition is that it's using biology to build useful things. It's really been underpinned by two major advances that are directly relevant to AI, which are standardization, which means parts, specifically microbes, DNA, even chemicals made by one lab can be used interchangeably by another lab. It's almost like the way screws have been standardized over the years, where you can use the same screwdriver head regardless of who actually manufactured the screw. As well as automation, and I think automation in this context, in this crowd that's tuning in, can mean a lot of different things to a lot of different people. To me, I usually think about lab automation, so that's robotics typically and high throughput screening. I've personally worked in 3 different biofoundries. So these are labs that are outfitted with this type of robotics that use them to iterate on the design, build, test, and learn cycle, which underpins a lot of engineering biology. I'm going to refer back to design, build, test, and learn because I think AI has a role to play for each one of those. But with lab automation specifically, so applying robotics to engineering biology, it's achieved this new scale, and I like to challenge people with thinking at that scale. So if you could, what would you build and test at a scale of 10,000 or 10 million? And these numbers are real. These are not just sort of made up back of the napkin. The experiments are happening at this scale now, where there are 10,000, 10 million, maybe more samples to keep track of. And that's obviously well beyond what a human is capable of keeping track of, even with the help of Microsoft Excel, which is, I think, a fallback for a lot of the start of experiments. We need something that's smarter. We need something better that can keep track of just to keep track of that data, let alone learn from it. But thinking about the design, build, test, and learn. So when it comes to builds, we're using robotics to move small volumes of liquid around, assembling DNA, purifying proteins, and culturing microbes. With testing, we're using this equipment for taking pictures of microbes, which for the Pytri folks is obviously very familiar, measuring growth, analyzing a chemical or protein output. Your eyes might have glazed over because these are all actually very repetitive tasks. And they sound maybe complicated at first, but once you can do it once, it's relatively easy to iterate on it. And as John alluded to, we can use automation both digitally and robotics, not to replace anyone's job, but to make our jobs better and freeing us to focus on the creative aspects of that work. I briefly talked about this presentation today with my partner, and she was like, "Well, okay, that makes sense. It's like a dishwasher in a restaurant, and specifically a machine dishwasher." And that analogy kind of stuck with me because it's true. These are repetitive but necessary tasks that we need automation's help with, and it frees the chef at the restaurant to focus on the creative aspects of cooking. And that's where I think automation, both digitally and robotics, can really help. And so I talked a little bit about the build and the test, but those being repetitive and tasks that can be iterated on. but there's also the design and the learn aspects of engineering biology. So with design, AI is already playing a huge role in designing proteins that are new to nature. I've started to apply some of that work in my own lab, and also for combing through genetic information and making sense of it all, and indeed predicting function or perhaps a disease state as a result of mutation. And then finally, learning. So it's one thing to be able to generate all of this data. So design, build, test, and learn, I think design, build, and test are becoming more routine, but it's learning from all of this data that we're now able to generate that is very challenging. And that requires us to synthesize data points, often many different types of data. So I've been interested in following Pytri's growth from that point of view, where they've expanded beyond colony counting. I'm not going to spoil the show or anything, but I think there's some interesting stuff they're going to share today about that. And then finally, informing the next experiment. So condensing and synthesizing data across tens of thousands to millions of time points and making educated guesses about what experiments to do next. So I hope that is beneficial information, engineering biology perspective, and I'm really excited to see where Pytri goes.
Thank you so much, Benjamin, for the insights offered. It makes me really proud to have him as a speaker today, because Ben was actually one of the very first partnerships we formed here at Pytri. But now talking about a more recent partnership we formed, it is my pleasure to introduce you to our research interns. Pytri has partnered with the University of Toronto to develop new data sets. And the first one that I will be presenting today is Xiao Ting, who will be speaking on our Salmonella dataset.
Everyone, I'm Xiao Ting, a research associate at Pytri, and I was responsible for the development of our Salmonella detection model. So Salmonella is a common type of foodborne pathogen, and it has several significant biological impacts. Firstly, salmonellosis caused by food contamination is considered as a major contributor to global food safety concerns, and that makes Salmonella detection a critical aspect in food testing protocols. And in terms of environmental safety, it's essential for the biosurveillance authorities and labs to monitor the distribution and spread of the pathogen in environments, like water sources and food production facilities. This surveillance is crucial for screening public health and controlling outbreaks. Additionally, quantification of Salmonella would also be used in many microbiology researches, like gastrointestinal disease studies. So considering its broad application, we think it's critical to include it as one of our datasets. And essentially, by automating tedious counting tasks, our Salmonella quantification feature will largely promote efficiency and accuracy for relevant labs and facilities.
Moving on, the next dataset introduced is Listeria, presented by Sharon.
Hello, I'm Sharon, and during my internship, I focused on Listeria detection. Listeria is a gram-positive bacterium and is of particular concern due to its widespread presence and impact on food safety. Listeriosis, the disease caused by Listeria monocytogenes, is a major concern. It's actually one of the leading causes of death from foodborne illnesses. Here in Canada, Listeria is a nationally notifiable disease, meaning health providers have to report cases to authorities for monitoring and control. The challenge with Listeria enumeration lies in its time-consuming and inconsistent results. Also, food companies must monitor Listeria levels to avoid recalls, which can damage their reputation and finances. This is where automated colony counting becomes a promising solution. It streamlines the process and can be beneficial not only for academic research, but also for food companies dealing with Listeria contamination. My project addressed these challenges by developing an automated method to count Listeria colonies on specific agar plates. One major challenge I encountered was the lack of data compared to other datasets. However, I overcame this challenge by using a similar agar type, which has the same colony and background color as the Listeria agar plate. Additionally, I use image augmentation techniques from RoboFlow to expand datasets. Ultimately, these efforts led to the successful launch of a new platform in Pytri, contributing to improved food safety measures. Thank you.
Moving on to the next data set, gel electrophoresis, which will be presented by Ash and Sarah. I think this one speaks to a lot of us that have been in the lab, as it is such a time-consuming process to quantify gel electrophoresis.
Hi everybody, so this is Team Gelectric, and it's composed of me, Sarah Varghese, and
me, Ashanafi Mandefro.
So I'll just get right into it. So our project is on the automation of gel electrophoresis analysis. So what exactly is gel electrophoresis? It's basically a really important technique used in research, such as molecular biology and biochemistry. Why do we use it? It's important for the separation of macromolecules like DNA, RNA, and proteins for later analysis. And how is it done? So these macromolecules are added to a gel matrix with an active electric field, and they're separated based on their size and charge. So what are the potential applications of GE? So Ash and I conducted a literature review on how GE can be used in research, and we came up with 4 main sectors, that being RNA analysis, DNA analysis, protein analysis, and environmental monitoring. So RNA analysis and DNA analysis are particularly important in research labs. So examples can be for gene expression analyses, microarrays, PCRs, or microsatellite analysis, SNP discovery, and DNA fingerprinting or footprinting. For protein analysis, it's mainly used in the pharmaceutical industry, so it's really important for the qualitative and quantitative assessment of purity and heterogeneity of protein samples. And lastly, for environmental monitoring, it can be used to identify whether there are potentially harmful organisms in local samples. So that could be microbes in water samples, and this can be done to conduct environmental impact studies to limit damage to ecological zones for research. So this is just a graphic that we've made of what gel electrophoresis could look like. As you can see here, this is just what the sample walls could look like. This is where you load your sample for analysis. Over here, this is just what the reference ladder looks like. And this is just a predetermined sample that you load to produce these predetermined bands. And you can use these bands to compare against your samples to determine their size. And this is just what a sample band would look like.
Yeah, and so the overall goal of this project was we wanted to first and foremost make things accurate and efficient in the sense that you have a machine learning model, you can reproduce it several times over, you're not open to human error. That idea that it's grounded in a math-based approach as opposed to an eyeball-based approach means you don't have the variation from scientist A to scientist B. You just run it through the model will always objectively sort of answer, this band is this far migrated. This protein is maybe this weight, this DNA molecule is this long. So objectivity is another one of those facets that we're trying to aim for with this system. And then also, as Sarah just explained earlier, DNA, not even DNA, gel electrophoresis as a whole is a very versatile tool. It's used in all parts of the biological sciences, as Sarah mentioned. And when we're automating this sort of system, we take away a lot of the time spent, a lot of the labor spent in the lab to just do this analysis. There's no discovery being done. We want to help impact this tool because of its versatile nature. But before any of that cool stuff, the automation happens, we had a lot of challenges that we sort of ran through, roadblocks, but we came through them. The first one was first and foremost finding images. In order to train an ML model and make it understand this is a gel, this is how far things migrate, this is where things migrate, you need to find images first and foremost. And finding images is difficult because, one, a lot of groups, they actually just publish their results. They're not publishing, this is a zip file of all my gel results. No, they're telling us, okay, the proteins we found are this heavy. The DNA we found is this long. We only see the statistics. So finding images, raw images, was very difficult. Luckily, we were able to find enough. And once you do find enough, you have to get to annotating them. So this is where you take just the raw image and it's like, you actually map to it. This part of the image means this sort of feature. We want the machine to understand this sort of part of the image, these arrangement of pixels means maybe this is a DNA band. As you do that several times over, the machine understands that, okay, maybe this is where a migrated band is. This is where the ruler is, things like that. But annotating it gets really tedious over and over again. And with hundreds of images, you have to do this several times over to increase and make sure that your model is reliable. And once all of that backend stuff was complete, Actually designing how we're going to implement this on the user-facing side was a little bit more tricky as well, just because gel electrophoresis has a fair amount of parts to it. There's things like you might have different rulers that you need to use, you have different sort of setups that you need to use as opposed to cell counting. So figuring out what's the easiest, most friendly way we can present this to the user, because that's the overall goal of Pytri. Automating these analyses, making it as friendly as possible, because Pytri is your personal microbiologist, after all.
That was just a brief overview of our project. If you guys want to see more, feel free to check out our website.
And this wraps up our datasets from the University of Toronto. A very important thing to note about the gel electrophoresis dataset is that it will be coming out on our website in a few weeks. And thank you to the University of Toronto. Now moving on to our research associates, Mika and Hamza. They will be presenting the blood smear dataset.
Hi everybody, my name is Mika Kasia.
Hi, I'm Hamza Abuzaid, and we have developed an automated system for applied microscopy peripheral blood smear analyses.
So, what is a peripheral blood smear? You go to the doctor, you get your blood taken for one reason or another. It goes through something called a complete blood count, and this gives you information from your doctor, the information about all of your red blood cells, white blood cells, platelets. Oftentimes, what's done alongside with that is something called a peripheral blood smear, and this can kind of help detect abnormalities. In pharmaceutical industry, this is typically used to monitor any possible side effects alongside just general progress if a drug is working or not. In academia, honestly, peripheral blood smears can be used for so many things, I can't list it. But in general, peripheral blood smears are really, really useful for counting red blood cells, white blood cells, and platelets, and monitoring for abnormally shaped red blood cells.
Thank you. Now for large industrial labs, what they typically do is that they use large, well not large, but expensive, fancy cell counting machines, which are great for, detecting white blood cell differentials, but they are very limited when it comes to RBC abnormalities. Now, what typically happens then is that often they switch to manual examinations of the blood smears. Now, for small to medium scale labs, such as ones in academia and other parts of industry, boy, they have it the worst. They do manual examinations by eye, through a light microscope, and do tally counting after staining, which is very tedious.
Now, for abnormal red blood cell types, there are many, and all of them have unique characteristics. But in real life, these categories aren't black and white. It really depends. Each hematologist, when someone looks at red blood cell, they know the category, but they might decide that this is one thing one day. Another hematologist might look at the same thing and say it's something slightly different. There is some variability, even though you have these categories. As well, counting blood cells by eye, it is prone to human error because of speed, could be distractions, or the very familiar feeling of exhaustion.
We all know it.
Yeah. So yeah, creating automated systems, it saves time, as you said, and eliminates human error and variability when it comes to identifying different cell types and RBC abnormalities. It also helps with precision, of results through consistent morphological, analyses and blood cell counts.
So the system that we created, our automated system, has the capability to give the normal numbers, the total red blood cell count, white blood cell, and platelet counts, but accessible for almost anybody. And what makes it even more special is that it can differentiate between 11 different types of red blood cell abnormalities. And this includes abelicytes, elliptocytes, spur cells. You got horn cells, spherocytes, stomatocytes, stigmocytes, etc., etc.
Yeah, that was a handful. So yeah, all you need to do to use this automated system is go to the Pytri website, upload your image, click on Analyze. Pretty simple, right? Once you do that, once it's analyzed, it generates a statistical report, which gives you, a total red blood cell, white blood cell, platelet counts, along with their percent concentrations, and along with a count of the different RBC abnormalities, all the types. Along with the statistical report, there will be an image, just like this one, and it gives you just a count of the red blood cells and the type of abnormalities there are. Okay.
And we are so excited to produce an accessible product that enables hematologists of all industries and lab sizes to be able to save time and increase precision. And if you have any questions about how we did this, how it works, how it can help you, please feel free to ask us during the Q&A session.
Thank you.
Thank you, Mika and Hamza, for this very impactful work. As they mentioned, after our event, you feel free to ask them questions about their data set. Last but not least, Ali will be wrapping up by telling us a little bit about the Petri dish capabilities that we have, including filtration membrane analysis and GFP fluorescence.
So the data set that Hamza and Mika were working on was an absolutely terrifying data set with 17 different types of entities to detect. I was not able to wrap my mind around how they were able to do that. So kudos to them. What me and Felix, our CSO, were working on were a bit more simpler data sets. Let's say that we had this goal of trying to automate everything that would take place in a microbiology lab or in a molecular biology lab. So for molecular biology, that would most likely be gel electrophoresis. When you look at a microbiology lab, more importantly, in terms of testing, you have quality control labs, which is basically anything that goes on a human or through a human needs to be tested for contamination. So think your cosmetics, think your shampoo, anything that goes through you or on you needs to be tested. And so for that, there are multiple ways. A lot of that is testing E. coli, a lot of that is swabbing, testing the products for contaminants. What you need for that is a total count of contaminants, which is something like this. So you have all these ugly types of fungi and bacteria on a petri dish. What you want is a total count of contaminants. So that is what our AI algorithm will be able to count. It's anything that is growing, quantifying that. Now, if you are testing your house for mold, fun fact, our houses are not very often tested for mold, but when you do get your house tested for mold or institute tested, what you do is you usually take a petri dish, you put it on the counter, you leave it for 30 minutes, you let the bacteria accumulate on that, and then you test it. And then you get your contaminants per minute. And so that's another big aspect of quality control. That's called a settle plate. So that's also something that looks like this, where you leave your petri dish out. Another aspect of that is testing our water samples for E. coli, for different types of bacteria and coliforms. What you do is you pass a membrane filter over water, you accumulate the residue, which is E. coli among other coliforms, and then you get a beautiful petri dish like this. And so you're going to want to quantify your E. coli and your other coliforms. It's also something that you need to do by hand. So you take your petri dish, you take your Sharpie, and you sit there counting them by hand. So we've worked on automating that, automating quality control labs through contaminants. And then the really cool one in molecular biology is GFP fluorescence. So you want to analyze your gene expression. What you do is you add a fluorescent tag to your gene, and then you see if it's expressed or not. If it's expressed, it's green, and you want a ratio of expressed to unexpressed genes in terms of colonies, and that's what GFP fluorescence is for. And so through all of this, we're trying to automate every type of quantification that you would do in a lab. It's something that takes a lot of time. It's something that we all face, all our interns face, and that we face in our daily work lives. And so we're working on automating that. So.
Okay, thank you so much, Ali, for telling us more about some of the data sets that we have here.